Well, thanks for the opportunity to share the progress that we're making at 11 Biotherapeutics. We are a public company, so I'll be making forward-looking statements and refer you to our most recent SEC filings for the most updated information. So at 11 Biotherapeutics, we're focused on designing and engineering differentiated protein therapeutics for ocular diseases. I'll be telling you a little bit about Isunakinra, our topical IL-1 inhibitor that's currently in phase 3 for allergic conjunctivitis. I'll update you on the progress we're making with our novel intravitreal IL-6 antibody and a little bit of update on the pipeline. So Isunakinra used to be called EBI-005. This is a topical, very potent inhibitor of IL-1 that's used for ocular surface inflammation, currently in a phase 3 trial for moderate to severe allergic conjunctivitis with an anticipated readout in the first quarter of 2016. We are also working on an intravitreally administered, a very potent, longer half-life IL-6 antibody for the treatment of diabetic macular edema and uveitis. This is a molecule that's currently in IND-enabling preclinical studies, and we anticipate filing an IND the first half of 2016, and then following on to clinical development. We also use our Rx platform to design and engineer differentiated proteins for other back-of-the-eye diseases, and right now we're working on a longer half-life VEGF molecule. So addressing the allergic conjunctivitis market, we want to make clear that what we're focused on is the moderate to severe patients who are not adequately treated with the standard of care antihistamines and mast cell stabilizers. There's about 11 million diagnosed patients in the United States, and about half of those patients are well treated with the antihistamine and mast cell stabilizers, but about half are not well treated, and these fall into the moderate category of about 4 million patients and a more severe category of about 1.8 million patients who frequently wind up on steroids, and we all are aware of the sight-threatening side effects of steroid therapy, especially in a chronic nature. So what's interesting from our perspective is that mechanistically, the more mild allergic conjunctivitis patients who respond well to the antihistamines and mast cell stabilizers are really suffering from what's referred to as the early phase response, whereas the patients who are suffering from more moderate to severe forms are really suffering from a more chronic inflammatory disease, which is not regulated by these preformed mediators such as histamine. So the market follows the mechanism. And when we think about this mechanism, when the mast cells degranulate, they do release these preformed mediators that drive the early phase response, but they also release cytokines and chemokines, which can drive this chronic inflammatory response. And IL-1 is known to be one of the most central modulators of this late phase inflammatory response in allergic conjunctivitis. And it actually both initiates and maintains this response. We wanted to design a phase 2 study that would mimic some of the preclinical work that had previously been done to demonstrate that an IL-1 inhibitor can treat more severe forms of allergic conjunctivitis. So we designed a clinical trial where patients were challenged multiple times with a conjunctival allergen challenge, treated for 2 weeks, and then challenged again. And the key here is that with these multiple challenges, we're driving patients into a more severe response. Additionally, we also were very conscious to enroll patients who had been known to fail antihistamine and mast cell stabilizer therapy before they were enrolled in the study, and this included even atopic patients. And so the data panel is demonstrating what happens in the early response, which is something that you see within about the first 30 minutes. You really don't get the same type of separation that we see when we look over 5 hours. Now, most of the conjunctival allergen challenge trials that have been described before are really measuring things within these first 30 minutes. But we've looked over a 5-hour period of time and have seen a separation between the drug in orange and the vehicle in gray over this entire 5-hour period of time. So when we're doing our analysis of our data, we're looking at this extended response time. And in this study, we were able to demonstrate a statistically significant separation between drug and vehicle at both the visit 6 and 7. So that's after that 5th and 6th allergen challenge in ocular itching. And also this was supported by statistically significant improvements in ocular tearing and associated nasal symptoms at these same time points. So this was the sort of fabric that we used to design our phase 3 trial. We wanted to pursue, want to pursue, a global registration plan. So both from that perspective and our discussion with health authorities, we decided that the best path forward was to study Isunakinra in an environmental trial. This is a trial that's ongoing right now. What we've basically done is confirm that the patients enrolled in the trial are known to be responsive to both perennial and a seasonal allergen. We did a assessment of whether or not the patients were sensitive to antihistamine and mast cell stabilizer therapies, and only enrolled patients who were not. We treated the patients over a 4-week period of time, and during that 4-week period of time, we will be measuring the primary endpoint of ocular itching as well as a number of secondary endpoints. And then finally, at the end of the study, a subset of patients will be rolled over into a conjunctival allergen challenge arm where they'll get 3 challenges similar to what we did in our phase 2 study, which allows us to confirm the data that we've seen in that phase 2 study. And again, this is a trial that we anticipate reading out the first quarter of next year. So moving on to diabetic macular edema, we believe that there's about 750,000 patients that are in the United States with DME, and we don't think that all of those patients are being adequately treated with the standard of care VEGF inhibitors. What we think is very interesting is to look at mechanisms that are different for DME, and we've been very focused on IL-6 because we know that IL-6 is upregulated in the patients who have DME, and we know that glucose is a very inflammatory molecule and can upregulate IL-6 levels. So when we thought about the unmet need in DME, we thought about the 25% or so of patients who are really not being well treated with the VEGF therapies. We thought about the idea that there are mechanisms that are upstream of VEGF, that if you could hit at that target, you'd be able to maybe hit both VEGF and non-VEGF driven inflammatory processes. We're very conscious of the fact that with VEGF, with IL-6, you need to have a comprehensive blockade, blocking all forms of IL-6 because it signals in a number of different ways. And that, as I think we think about for all retinal therapies right now, a decreased frequency of administration would be very important. So a long half-life antibody would be important. And that's what we've done in engineering the anti-IL-6 antibody EBI-031, is really hit all 3 of these sort of categories. So again, as we mentioned, IL-6 is actually upstream of the production of VEGF, so vascular leak driven by VEGF could potentially be treated with IL-6 blockade. We did a choroidal neovascularization where we blocked with both an anti-VEGF and anti-IL-6 and see similar response in the CNV models. But we also know that IL-6 is upstream of the Ang-Tie pathway and direct inflammatory vascular leak mechanism. So we believe that IL-6 is a more comprehensive way to treat diabetic macular edema. We're also very encouraged by the data that's recently been reported on the use of systemic tocilizumab, which is an anti-IL-6 receptor antibody that's been effective in treating the cystoid macular edema associated with uveitis. So all this put together makes us very encouraged about our IL-6 program. So EBI-031 was specifically designed and engineered to be extremely potent. So this is a femtomolar potency antibody. And when we put that potency together with the longer half-life that we've seen in animals, both rabbits and monkeys, compared to different VEGF inhibitor antibodies or receptor traps, we put that together in a modeling, and we can demonstrate that we could get 95% blockade of IL-6 over a period of at least 120 days, if not longer, which should translate to a much reduced frequency of administration relative to the VEGF antibodies or blockers in DME, and possibly give us a frequency of administration of not more than once every 3 months. So we're very excited to be moving our IL-6 antibody towards the clinic and anticipate the IND in the first half of 2016. We'll have our readout for Isunakinra and allergic conjunctivitis in the first quarter of 2016, and then if the data from that study is favorable, moving into phase 3 the second half of 2016. Thanks very much.